Hey guys, today we're going to talk about DNA evidence and how it can be used to solve crimes in forensic science. So here we go. All right, so you've probably studied DNA in other courses like biology, AP biology, maybe you've talked about it in chemistry. It's a very large molecule and it stands for deoxyribonucleic acid. That's what the actual acronym is. It contains all our genetic information, which is on chromosomes. And I know that you guys have learned about that, like I said, in biology, but I'm not going to go huge into that. But I do want to talk about some different things that make up the DNA. So as we look at the structure of it, we say that it's a helix, a double helix molecule, and that it's made of sugar molecules and phosphate molecules. And there's different base pairs that go together, and you can see in this picture here, the C goes with a G and the A goes with a T. Okay, and the A is the adenine and T is thionine. And there is our adenine. There's our phosphate backbone. There's our thymine. There's our cytosine. There's our guanine. And nitrogen bonds are in between them. Okay, just wanted to show you the structure of it and talk about that real quick. So how do we actually use DNA as evidence? So most of us would say, oh, okay, it's to match a suspect from evidence found at a crime scene to a suspect, right? It's not always used for that. It is used for that for sure, but it also can be used to figure out if there's a mass disaster and you want to figure out like who were the people that died. You know, maybe there's a plane crash and we're trying to figure out, you know, can we find out who actually died in the plane crash? And they'll look at the DNA and figure out, you know, who was there. Or maybe there was some sort of fire and they don't know who was in the house at the time. So they use DNA in that respect. The other way that they do it is they're trying to link more than one crime scene to each other, right? So you get DNA from this crime scene and then you see, oh, this guy was a part of that crime as well. And then, you know, that's how it's linked. So that's another way that we can use it. I was just trying to think if there was a case where a guy hid in the back of a girl's car, and it was she parked at like a Starbucks to study, and for whatever reason it was an electric car and she left it running. And when she left it running, the guy jumped in the back. And then when he jumped in the back of the car, when she got back in the car, he strangled her and raped her. And they were able to find his DNA later. They originally thought it was her boyfriend that did it, but then later they found his DNA at another crime scene, and then they were able to link it. And that way they were able to say, okay, this isn't the boyfriend's DNA, and this DNA was also at another crime scene, so somebody else is involved here. So that's an example of that. It can also, so, you know, anytime there's a crime, they interrogate the suspects and they ask them, like, what happened here? Or why were you at this location? And so a lot of times they're using the DNA to corroborate the person's story or say, hey, you know, you said you weren't here, but your DNA put you here this time. Which, again, like we said with the fingerprint, it's wild. We can tell, like, how old the fingerprint was, you know, in a matter of hours. So it'll put them in a certain place at a certain time. It can also, so a lot of people are like, oh, I have DNA under my fingernails. So clearly that crime was self-defense. Was it? So there's two ways on that. You know, it could be self-defense or they, you know, yes, but that doesn't mean they didn't start it, right? So that's another way they can use it. All right. So a lot of people say that DNA evidence is better than other evidence, and here's why. So first of all, we have DNA everywhere on us. And now that we can use such small little trace amounts of DNA for analysis, it's even more helpful because anytime we touch something, it's Lockhart's exchange principle that we're going to leave something behind, right? So that we can get DNA all the time, which is helpful because, you know, whether you leave hair or whether you have a fingerprint, there's going to be DNA. DNA lasts longer than some of the other specimens you might collect. And it can indicate if people are related, right? So you can see if that was somebody that was a sibling or a parent or a child, and if they're actually related. So like that would be happening in like a paternity case. You guys have seen like those talk shows where people get up there and they're like, "Oh, that's not my kid. That's she was with another guy," and that's when they do a paternity test to see who does the kid actually belong to. That would be in that case. They're looking at the DNA there. Other thing is DNA doesn't actually mix. So if we have different blood types can get mixed up, and then we can't tell what's what. But DNA doesn't do that. Strands of DNA like don't mix together. So once you have a strand of DNA, if there's another strand in there, it's from somebody else. But a DNA strand will tell you specifically who that person is, and then another strand is somebody else. Whereas blood, you could have somebody that has the same blood type, right? And then also you could have blood types that mix, and then you get false identifications. So you don't want that. What else did I need to tell you guys about this? I just want to forget to tell you something. Okay. All right. So what factors affect DNA evidence? So in Florida, we have a terrible environment for keeping DNA evidence because the big things that mess with DNA are humidity and heat. Guess what? We have both. So it is really important that you need to figure out, or you need to keep your DNA evidence in a cool, dry storage. So that's the big thing. So we never package DNA items in anything plastic because, as you know, if you're package something in a plastic bag, it'll get condensation on the inside because of the moisture. Okay, so in this one it says DNA testing cannot identify when the suspect was at the crime scene or for how long. It depends on what you have. So like I said, for your fingerprint evidence, you can tell based on the fingerprint, how long somebody's been there. But, if it's just a piece of hair, you really can't tell that. So that's what that's talking about there. All right, CODIS. You guys have probably heard about CODIS on crime shows. It's literally just the database of where DNA is stored. So it's only from people that have committed a crime, right, or they've been arrested. Also, there's people in there that are military personnel and work for the government, right, for different things, because their fingerprints, they had to get fingerprinted before they started their job. It's also missing persons, are put through CODIS. Here's the problem. When is CODIS not helpful, or what's the main drawback of it? Most of our DNA is not in CODIS. So if you've never committed a crime and they put your DNA into there, they're not going to find anything, right? So that's like the biggest problem with it. And there's just some information. So most of our DNA is similar, right? That's what it's saying. How much is unique? 0.1%. That's why DNA differentiates us so well, that only 0.1% of it's different. So if yours is different from somebody else's, then obviously it makes a better chance that it identifies a person. All right, so different sources of DNA at the crime scene. All right, there's at least 10 that kind of a list that a CSI would look for. So take a minute and see if you can think of a couple in your head, and then I'll go through these. You'd be surprised. I don't think you guys realize all the places that DNA would be found. Okay, so let's take a look at them. So obviously the pictures there are giving you some indication of some hints. All right, so the picture on the left top is maggots. And remember, if maggots are feeding on a body, and then when we come to the body and it's just bones, we can actually use those maggots to analyze them for DNA because they have the tissue inside them, and they can get DNA from that, which is crazy. And then you can actually, from a ball cap, you can get sweat residue, and they'll do that a lot. They can get them from skin cells from a strangled person. They're talking about, so if somebody strangles you with their hands, they can actually get skin cells that were on the hands on the actual person's neck. They can get them from that. And you can see other ones. For the hair, the root has to be attached for the hair to have DNA. So that's something to note as well there. Some other things that have DNA that you may not think about is bones, teeth. Again, you have to have the root of the tooth in there, but definitely bones. Your urine actually has DNA in it, shockingly. and fingerprints and skin cells. Okay, so there's actually a checklist that the National Institute of Justice says for CSIs to look at when they go to a crime scene. Here's stuff they should check for DNA. Right? So I wanted to show you this real quick. I want to show you this website. So look at the different things. It's very interesting. Yeah. So they collect bullets, they collect any used condoms, they collect any tapes, ropes, cords at the scene, any used stamps or envelopes. Because remember, you have to lick the stamp, so you're going to have DNA from that, from your saliva. Hats or headgear. Any of you guys had to use headgear when you had braces? It's that thing you put in your mouth. So that's what they're talking about there. Also, headgear could be any kind of helmet of any sort. Obviously, glasses or even sunglasses would have DNA on them. Cups that had been previously used, that's not on here, but that's another one. Cell phones are covered in gross stuff. You can see them swabbing it. Cell phone there. The cigarette butt as well will have saliva. I accidentally put an N there. It's everything in the bathroom garbage. So they would look at a lot of stuff because a lot of that stuff would have DNA on it. If you blow your nose. That was a case I was listening to the other day. It was a crime junkie we didn't do. And that's how they were to catch the guy, because he blew his nose and threw it in the garbage at a hotel. And they were able to get his DNA from that tissue that was in the garbage. And they said that the judge made that allowable because the crime scene investigators didn't, set him up for that. Like it was something he willingly threw away. Okay, I want to show you guys this website because it has some cool questions. So let's say you're at a crime scene and it says, like, look at the first question here. What was the blue hooded sweatshirt found by the police probably the one worn by the bank robber? So if the answer is yes, then that's good because you can analyze, right, the hooded sweatshirt. If not, then you don't even need to look at the hair analysis to help identify the robber. Number 2, did the FBI correctly identify? This is the mitochondrial DNA, which we're going to get to in a second, but that's the DNA that is in the mitochondrial in the cell, mitochondria, and it only gives us the maternal side. That's something significant. Sequences of the substance of the suspect, sweatshirt, hood, and known defendant's hair samples. So yes, so you're going through all these checklists, and it helps you kind of get to like narrow some things down. How do we figure out who this person was? So I just kind of want to show you that, see what it looks like. Okay. All right. So this is kind of new in the world of DNA, new-ish. Some of you guys may not have heard it before, but so the first thing it says is DNA unique to everyone. So when you think about it, everybody's like, yeah, it has to be, right? Okay. So the answer is no. There's one exception, and the exception is twins. So identical twins, when they are first born, their DNA is identical. Something people may not know, and you may not know this by yourself, is there's a subtopic of DNA called epigenetics. So epigenetics means that as we get older, our DNA mutates based on different things we're exposed to. So for example, let's say that we have 2 identical twins. When they're first born, they look the most alike they ever will. Because as they grow up, they are subjected to different environments, right? Maybe one of them likes to play sports. Maybe one of them really eats unhealthy. Maybe one of them starts smoking or using drugs. Maybe one of them, the parents is nicer to. All of these things affect our health. And that's called epigenetics, which means that different things in the environment, pollution, depending on like where you live, all of these things can modify our DNA. And therefore, you can actually find differences in identical twins' DNA if you know what to look for. Okay, so that's something that is kind of a new study in the DNA world. Okay. All right, so when we're looking at a crime scene, what we're actually looking for once we analyze the DNA, this is like the lab we did, we could do like a PCR analysis and get this is what the gel looks like when you run a gel. And so this just shows you the different markers at the locations of where the DNA is. And so if the markers line up, then you say, oh, okay, that looks like a match. If they don't line up, then it's definitely not a match. And sometimes they'll match up in some parts and not others. And that could indicate people are related, okay? But it has to be the same individual. All right, so when DNA analysis first started in the early 80s, we had a RFLP technique, and I'm going to go through each of the techniques in a second. But I want you to take a minute, pause this, and watch this. Actually, we can watch this video here. I'll watch it. These are, but I'm going to go through these in a second, these different types. DNA has progressed, the analysis of it has progressed significantly from RFLP to what we use now. And right now we use a lot of PCR, actually. That's the one that most people use. And the big thing about it is we've now gone from being able to use, having to have a big sample of DNA to just a really tiny bit. And that's made a huge difference. Okay, so let's take a look at this video here.
It really scares me when a politician like Joe Biden says he's going to raise our taxes.
Your taxes are going to be raised, not cut.
Joe Biden is literally going to raise taxes on us. So our American way of life is at threat with Joe Biden.
Hi, it's Mr. Anderson, and in this podcast, I'm going to talk about DNA fingerprinting. We sometimes refer to this as DNA profiling or your genetic fingerprint. And basically, it started with this guy, Alec Jeffreys. Basically, in his lab, he was working with X-ray and looking at DNA. And what he figured out is he could tell a lot about a person by looking at their DNA, quote unquote, fingerprint. In other words, he could see who they were related to or who they weren't. He could tell paternity, for example. And so he was working at the University of Leicester and basically figured out this whole idea of DNA fingerprinting. This is around 1984. And basically for the next 3 years, all DNA fingerprinting on the planet went through this university. And so eventually it was privatized, and this is everywhere. And it'll probably eventually be replaced by just DNA sequencing all the letters in DNA. But to make it understandable, essentially what we have in a human is we have long linear segments of DNA, but within that we have these genes. And so 99.9% of our DNA in everyone is going to be exactly the same. The genes are going to be the same, but you're going to have different copies or alleles of those genes. That's what makes you, you. But if we look into this area in the middle, we used to call this junk DNA, but now we know it's really important in controlling gene expression. We find that there's quite a bit of variability in here, which shouldn't surprise us because the gene makes the protein, and the protein makes the phenotype, and that's really what natural selection is selecting for or against. But this in the middle can go crazy, and so it does. And so an example of one that we use in DNA fingerprinting is something called short tandem repeats. Originally, we started with something called VNTR, variable number tandem repeats, and you'll find in DNA sequencing that you have all kinds of. So we had STRs, we have VNTRs. Before that, we had restriction, fragment length, polymorphisms, and so there's a bunch of different things that we could look at. But we're kind of moved to this idea of these short tandem repeats. They work great. There's quite a bit of variability in individuals. And so what is it? You basically have letters of DNA that repeat over and 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 over. So sometimes, 50 times it repeats. And so what does that look like? Well, if we have these three individuals, we'll call this Mr. Blonde, Mrs. Red, and then Mr. Mustache. And so if we look at these three people, their genes are going to be the same, but these STRs are going to be different. These single or short tandem repeats are going to be different. You can see that Mr. Blonde has more than Mr. Mustache and less than Mrs. Red. And so if I make that a little bit easier to grasp onto, if I count them out and then remove everything else, what we get is variability between all individuals. Everything else was the same, but we see variability in here. And we can cut these sections out using restriction enzymes, and then we can amplify them using polymerase chain reaction, and then we can separate them using gel electrophoresis. So how does that work? Basically, I'll take the DNA and I'll put them in a little well. And so we're looking down on this. And so this is a agarose gel. I could put Mrs. Red's and then Mr. Mustache. I could put those all in DNA. basically I would then turn on the voltage. So there's going to be a positive charge here, negative charge up here. DNA is a negative charge, and so it's going to be pulled towards the positive. And so what's going to happen is those little fragments of DNA are going to migrate. And so what does that allow me to do? It allows me to tell the difference between each of these individuals. And so this is their fingerprint. But you can tell that this is a really bad fingerprint because we've got these two are exactly the same here. And so when they really do DNA profiling, what they do is they generally use 13 different sections like this. And then those 13 sections are each going to be highly variable. So it's a good way to tell who's who. When would we ever want to do this? Forensics is one reason, and then also in maternity, figuring out who's dad. And so let's talk about the murder. There was a murder that was committed. Somebody was brutally murdered by one of these three suspects.
Okay, so look at the three suspects. Just take a guess, just based on what they look like. Which one do you think is the murderer? Okay, I just want you to look at that real quick. All right, now we're going to look at their DNA.
So Mr. Blonde, Mrs. Red, or Mr. Mustache. But they left blood at the scene. And so what I can do is I can grab samples of DNA from each of our suspects, and then I could grab the blood itself, and then I could do DNA fingerprinting. So before we separate them, you may think to yourself, which of these looks guilty? Who looks like they're capable of murder? And if we separate them then using that gel, what we can see is that Mr. Blonde is guilty. In other words, his blood matches up with the crime scene. And so what do I mean by matching up? Well, those single or those short tandem repeats, if we look horizontally, are going to be exactly lined up. And if we were to look at Mr. Blonde's son, we'd find more similarities than we would between the others. And so basically that's DNA profiling, DNA fingerprinting. It's much more sophisticated than that, but again, it's kind of on its way out. We'll eventually replace this with DNA sequencing. In the US, the FBI has started creating this database of DNA, which is a little scary. And basically what they use are 13 different areas within the chromosome or the genome, and then they're looking at those short tandem repeats in there. Now, why do I say that's a little bit?
Okay, so one thing that I want to point out is he thinks it's scary that we're looking at DNA and keeping, all these records. A lot of people are like, oh, why? That's a good thing, right? Because we can catch criminals. But the drawback of it, I can tell you what people are stressed out about and worried about is if they have your DNA on file. Have you ever seen on commercials where they say, oh, test your DNA to see if you're predisposed to get cancer, right? Like if you're somebody that's more likely to get breast cancer, or if you're more likely to get muscular sclerosis, whatever. I could never do one of those tests, by the way, because I would be panicked about it the rest of my life. But that is a reason that DNA being on file for everybody makes people nervous, because guess who could use that information? Insurance companies. So if you're going to sign up for insurance, your insurance prices can be super high if you're somebody that's predisposed for any of those conditions, even if you don't currently have them, which that's what people are nervous about. All right. So here are the different, I'm going to just show you here the different types of DNA analysis. So you can see this one's the restriction fragment link polymorphism, or RFLP. This is one of the original ones. Again, the reason that they don't use this anymore is that you have to have a lot of DNA to use it, and it doesn't do well with DNA that's a little old or degraded, which AKA we have in Florida because of humidity and heat. You can see the process down below. The process is similar for a lot of them. But look at the sexual assault case I have on the bottom right. So look at suspect 1 and suspect 2. Okay. And here is the victim. Okay. Who does it look like it lines up with? Suspect 1 or suspect 2? Or neither? Click it lines up. Victim and female cells, that's what it should be. And then the boyfriend, let's see, what does it line up with? That's the control. It's not the boyfriend. this right here. It's the sperm, but it does not line up with suspect 1 or suspect 2, right? Oh yeah, it does. Sorry. Suspect, oh, I didn't mean to click on it. Suspect, I was looking at the wrong column. Suspect 1 is who it is because that's the sperm evidence. Okay. Sorry, I was looking at the wrong row. Okay, PCR, like I said, is what is used today. The reason it's so important is that we used to have this much blood, which, is significant size. In comparison, we can have a little pin drop of blood and actually get DNA from it. So that's huge. It's quicker to do the analysis. It's also cheaper to do, and it gives us, a good DNA analysis. The only issue is that it's easy to contaminate because you have such a small sample. If it's a small sample like that, it's easy to contaminate it. All right, so let's take a look at these. So who did it in the first picture here? So our blood stain is the evidence, and who does it looks like matches up, or who, like more than one? Okay, it looks to me like it's got to be suspect 3. Right. Okay, who's the daddy? Which sample is most likely to be father? So remember, the child can have lines from the mother and the father. The M up here is the mother, and the F is F1 is a father and F2 is a father. So here's a child that matches up with father 1. Here's a child that matches up with the mother. This is a child that matches up with father 1. It's got to be father 1, right? Okay. And then look at these pairs, these sets of the gels that were run. And are these twins? So we're looking for, again, we're assuming that they're young, at least. They haven't changed a lot as far as the DNA goes. And by the way, most of the DNA will be really similar. It's just going to be very little changes that the epigenetics will change. So it looks like B and C and maybe D. Nah, probably not D. So just B and C are identical twins. Okay. All right. You saw this in the video that you showed, the points that they put into CODIS. They look at the DNA to put in there. Those are the 13 specific points. It's been used, you can see, to identify, the World Trade Center terrorist attacks. That's what they use to do that, those different markers. Also, interesting, you may not realize this, but they also use it to monitor, they use the DNA to monitor how transplant patients are doing. If somebody gets a heart transplant, they can, see how they're doing based on that. All right, mitochondrial DNA, again, mitochondrial DNA is good because it lasts a long time, and it also lasts through some sort of trauma. For example, if you've been in an explosion or you've been in a fire, the mitochondrial DNA can still be harvested from bones, from the mitochondria. And the only problem with the mitochondrial DNA is it only gives the maternal side. So that can be, if you have several sisters, well, they were all related to the same mom. So that makes it harder to determine who's who. We don't need to look at that. Aside from typical matching to a suspect of a forensic case, what are other common uses for DNA analysis to identify people? So we've talked about this, but for some of them. So some would be mass disasters, right? Forensic anthropologists might, send some DNA samples away to be analyzed because they want to know who were these people that died in that pit. Like that guy that we saw that did the TED Talk that was talking about the people that were all in the mass grave. That's how they identify who they are. People that are missing persons, right? And they have bones. They're trying to figure out who it was, who it is. We already said before, a paternity test. That's another reason to figure out who's the dad, stuff like that. Okay, so again, here is another picture of the markers they're using to look at in CODIS to figure, who they would put into CODIS and like what parts of the DNA they submit. And everybody at those points, they find those statistically significant that at those points, people's DNA is different. That's the point of those. Okay, so let's go through and look at these statements and which 3 statements below are true. Okay, so read through them really quick and see if you can figure out to yourself what they are. We didn't really talk about the last one yet, but we will in a minute. Okay, so I'm going to highlight the 3 that are true. Okay, so remember, to have the same DNA, it'd have to be identical twins. And again, that's only really the same at the beginning of their life, and it changes throughout. And you know, third one. Okay, so I want to show you this little video, and you can pause this and open it in another thing if you want. But let's go to this because there's some questions I want to ask you about it. This case is very annoying to me because it's not solved. Sorry to ruin it already.
It was one of the most sensational murders of the century, and it was never solved. Marilyn Shepard, the wife of a handsome surgeon, killed by more than 15 blows to her head. Dr. Sam Shepard was convicted of murder in a highly publicized trial. Sentenced to life in prison, he served 10 years until the U.S. Supreme Court overturned his conviction. In a second trial, Shepard was acquitted, but suspicions about his guilt remained.
My mother was murdered. I want my mother's murder solved.
Now the son of Marilyn and Sam Shepard is taking the case to court one final time, suing the state of Ohio for $2 million for the wrongful imprisonment of his father. Gilbert needs to convince a jury beyond all reasonable doubt that Shepard did not kill his wife.
Unless we do it on rebuttal.
He'd like to use a powerful new scientific tool, unavailable at the earlier trials, that could potentially settle the question once and for all. Forensic DNA analysis. Human DNA is made up of billions of chemical building blocks. At certain locations, the arrangement of these building blocks varies in predictable ways from one individual to another. These variations are called alleles, and they are the key to DNA fingerprinting. If enough of them can be observed and compared, these genetic variations can identify an individual as uniquely as a fingerprint. Scientists are now able to compare the DNA found at crime scenes to DNA taken from suspects. As valuable as it may be in prosecuting crime, it's DNA's ability to rule out suspects that holds the most power. Gilbert enlists the aid of Dr. Mohammed Tahir, a forensic DNA specialist in Indianapolis. He sends Tahir a set of artifacts, all stained with blood from the crime. Among them, a wood chip lifted from the stairs of the Shepard's house, a section of flooring from the porch. Tahir chooses a test called DQA1. The DQA1 test is designed to work with tiny quantities of DNA, but it isn't very discriminating. The test can identify only 8 different alleles in 42 possible combinations. His first step is to increase the quantity of DNA the test will have to work with. To do this, he places a portion of each crime scene sample into a vial. Then he adds a chemical cocktail that stimulates the process of DNA replication. The samples are then placed into a thermal cycler, which speeds the process of replication through precise warming and cooling of the samples. Within hours, any DNA present in the original samples has replicated into millions of copies. He obtains Marilyn's DNA from a few strands of her hair collected after the murder. Finding Dr. Shepard's DNA proves more difficult. So Sam Reese Shepard grants permission to have his father's body exhumed for tissue sampling. Dr. Tahir can now complete the testing. The DNA from each of the samples is now poured over the sensitized paper test strips that will reveal the long-awaited results. Each test strip is printed with a set of numbers. The numbers refer to the 8 specific alleles the test can detect. Indicators turn blue when the test has detected a specific allele. In this example, the test has detected the number 2 allele and the 4.1 allele. Once Tahir obtains a test strip for each DNA sample, he will carefully chart the results and present his findings to the defense team. At the first trial, the prosecution argued that the blood trail came from a weapon dripping with Marilyn's blood, carried by the killer as he fled the house. What does the DNA show? The porch stain tests positive for the 4.1 allele only. The wood chip also shows the 4.1, as well as the alleles 1.1, 2, and 3. How does this compare to Marilyn Shepard's DNA? Her alleles turn out to be 1.1 and 1.3. One or both of Marilyn's alleles are absent from the blood trail samples. Marilyn is therefore excluded as a donor to these stains. So the prosecution's theory was wrong. A murder weapon dripping with Marilyn's blood didn't leave the blood trail. Then whose blood was it? Was it Dr. Shepard? His alleles turn out to be 1.2 and 1.3. So Dr. Shepard is also excluded from the blood trail.
Okay, so let's look at some questions that deal with that. Oh. All right, here we go. All right, choose the best answer for each. Number 1, who is the victim? Hopefully you got Marilyn Shepard. What are the keys to DNA fingerprinting? You said B, you were correct, the alleles. Where did the scientists get the sample of DNA from Marilyn Shepard? Her hair. Remember, the fingernail was for Sam. Whose blood was found in the blood trail? Neither. So whose blood was it probably? It was probably the suspect, whoever committed the crime, since it wasn't Marilyn's and it wasn't her husband's. It had to be whoever committed the crime. But again, they don't have CODIS at that point, right? And, there's no way to figure it out. They don't have any other leads. Okay, so number 1, how was DNA evidence used to prove that Sam Shepard did not murder his wife? And why wasn't this evidence used when the case first came to court? Well, the reason it wasn't because it wasn't available, right? The case was a long time ago, way before we used DNA analysis. And so they were able to show that the actual alleles didn't match up. Why do you think the DQA1 test was chosen for DNA analysis in this case instead of another more powerful genetic test? Well, 1, it was probably one of the ones that they had then, number 1. Number 2, they said it wasn't discriminatory, but it was a powerful test. So the results, and what they were trying to do was they were just trying to figure out if it was Dr. Shepard or not, really, right? And so that did the job of that. If the blood trail left at the murder scene wasn't Marilyn's or Sam's, whose blood might it have been? Okay, so sorry, I already answered that, but it was probably the suspect. If you were a juror on this trial, would you be convinced by the DNA evidence? So to be honest, if it was me, I'd want to see some more now with the better DNA tests we have. I'd like to see it not just test for those few alleles they were looking at, but do more like we do now. But I probably would have at that time, if I had to rule on that, I would definitely have agreed based on DNA evidence they gave, because they were able to exclude Dr. Shepard. And again, we don't have another suspect. Okay, This is not what we're doing out of class. All right, so after this, I need you guys to work on the module 9 study questions dealing with DNA. Make sure you look through the modules as well, like read through them because there's more information on DNA there. And that's it. Have a great day, guys. Excellent.